Okay. Okay, so I'm very pleased to have our guest today, Dr. Alan Love. Dr. Love is a philosopher of science whose research concentrates on conceptual issues in the life sciences. He uses a he uses a combination of approaches, collaborating with scientific practitioners as well as historical investigation to analyze developmental biology, evolutionary biology, evo-devo, functional morphology, molecular biology, genetics, paleontology, with special attention to epistemic challenges arising from interdisciplinarity. So that's a broad scope, and it's outside of our regular type of seminar, but I think it's really going to be useful for us, especially across the range of systems that we represent here. I got to know Dr. Love quite some time ago now when I was a graduate student at the University of Minnesota, which is where he's based, and he's a distinguished McKnight professor and the director for the Minnesota Center for Philosophy of Science. And maybe I shouldn't say too much about what I'm hoping this will do because I don't want to unfairly burden the presentation. But I think that this will help us learn from each other more effectively as well, and to think outside of our little box of our own project. And I think those are pretty central goals for the EvoFunPath program, bringing together such a diverse range of topics and disciplines as we do. So let me just give you a couple more details about Dr. Love. So he's earned his PhD in History and Philosophy of Science from the University of Pittsburgh in 2005, prior to which he also earned master's degrees in both biology and philosophy. And I've had a few discussions with Dr. Love leading up to this to talk about, how this might be most useful and for him to understand our group. And so I'm really appreciative that you were willing, Alan, to put in that upfront work as well and to personalize this for us. So I'm very excited for your talk, and I'll turn it over to you.
Thank you, Matt. Appreciate it. Happy to be here. And I guess I can say at the beginning, since you now know that this was Matt's initiative, that any and all criticism can be directed to him afterwards. No, just kidding. So let me go ahead and share my slides, and we'll get going. All right. Is that coming through okay? People able to see the slides? All right. Great. So what I want to do today is talk about some sort of conceptual issues that are at the back of much of modern biology. And I am hoping that that conversation can be specially tailored to the needs of the EvoFunPath group. And so we'll sort of move in that direction over the course of the talk. I want to start with a little historical perspective. This book, Advice for a Young Investigator, was by Spanish biologist Santiago Ramon y Cajal. And in it, he laid out prerequisites for those new to scientific inquiry. So, for example, in his chapter, What Newcomers to Biological Research Should Know, he argued that specialization, mastery of technique, and original data were essential. So things that maybe are exactly what you would expect. But he also advocated for the importance of general education, including philosophy to learn guiding principles of inquiry. So this is a quote from that same section. More than anything, the study of philosophy offers good preparation and excellent mental gymnastics for the laboratory worker. And in later chapter, he marshaled historical examples to help those he was working with kind of illustrate how observation, experimentation, hypothesizing, and testing can be done well. He was not advocating a formal curriculum in the sense that he was saying everybody should somehow take history of science or philosophy of science classes, but rather he was giving some principles for how to provide informal ongoing guidance to new investigators while pushing them to be better scientists. And that mentoring clearly remains crucial today for both graduate students and postdocs. However, the study of history or philosophy by scientists today does not typically play a role in shaping the next generation of biologists. And yet I think that if we do incorporate some of that, then we position future scientists to be in a better position to not only interpret their own practice, but also to reflect on it and foster creativity and innovation. I can say more about that in Q&A. We've got a current paper under review about this that should be coming out relatively soon in Proceedings of Royal Society B. But today I want to focus on how this shows up in the context of model organisms, the kind of heartbeat of contemporary biology, and in particular, the way in which those models inform our ways of thinking about general knowledge and what counts as generality in how they represent life and the properties that biologists want to study. So here's what I want to do in the time that I have. I want to start by talking about model organisms and their prevalence. Then I want to talk a little bit about how we can distinguish different kinds of models and on what basis. And then transition to talking about generality, why it's important, and how we can seek and find generality in the context of our preferred models and why we might want to do that. So we'll sort of see a natural break point here between the first half, more about models and model organisms, second half, more about generality and recommendations for what to do. So why are model organisms so prevalent in biology? Well, unlike electrons or quarks in physics, biology is faced with a massive amount of diversity in its basic units. Organisms and species and their component parts and traits. Given the limits of time and money, biologists utilize only a small number of species to experimentally elucidate various properties of organisms and species. And those experimental models permit researchers to investigate particular phenomena in great depth, facilitate the detailed dissection of causal relationships through experimental intervention, and other kinds of modeling endeavors. And many of you might be familiar with the fact that these are advocated by funding agencies like the National Institutes of Health here in the United States, but also in other agencies around the world. That work that is done in this small subset of models is oftentimes supported because there are communities and stock centers and standardization that has grown up around them to support deeper and more informative analyses using lots of diverse methods. Now, the common justifications for model organisms, one is that conserved genetic or cellular mechanisms are there, and that if we've discovered them in one species or organisms, then we actually are gaining some kind of insight into others. That is, of course, an interesting empirical matter that has emerged, especially over the last 50 years, but it's clearly part of the justification. Second is medical applications. So we obviously can't experiment on humans, so we can use model organisms as the basis for discovering causal details and possible interventions for diseases through studying those different kinds of models. And then finally, we might justify our model organism based on a particular set of questions that we're asking, where that model is either historically or in a way that is relatively new, helping us get at those particular kinds of questions. And obviously, there's a diversity of questions to which we might think some models are better suited than others for informing us about them. Now, if we want to sort of dig a little bit deeper on sort of what the basis of model organisms is, we can sort of see it reflected even here in this NIH page. It's a little bit out of date at this point, but reflects the basic rationale. One is this idea that we can extrapolate from the few species we look at to others, and that they are in some ways practically useful to do experimental work on. So I want to talk a little bit about those two dimensions. Representation, how a model organism represents other organisms or species, and also manipulation, how practical benefits to organisms support their choice. So in thinking about representational criteria for model organisms, we can think about the scope of that, how widely into what biological systems the results and lessons learned are projected. So if we are studying zebrafish to learn about vertebrates, then zebrafish as a model organism is supposed to have vertebrates as its representational scope. And then, of course, we can have targets. What biological phenomena are explored by studying the model system? So if the research group is using zebrafish to explore how neural ectoderm becomes hindbrain, then nervous system development is the representational target. And as I've intentionally set up here in the example, you can see that in many cases, the choice of the representational target is tightly connected to the representational scope. So studying zebrafish nervous system development to learn about vertebrate nervous system development more generally. Now, a couple of distinctions are helpful in this space when we're thinking about the scope and target. One is the difference between mechanisms and phenomena. So mechanisms is a familiar term, causal interactions that underlie the trait or property of a system. And then the phenomena would be the trait or the property of the system. And when we think about scope or we think about target, we can be either thinking about mechanism or phenomena or both. We also could be thinking about specificity versus variety. We could be very interested in whether there is a single mechanism that our model represents or a phenomena that is distinctive. We might think of that as the right organism for the job. But also, we sometimes want our models to do multiple things and choose them for that basis, a more general purpose type fit with respect to scope and target. So we can represent this kind of in a possibility space here by sort of showing that in thinking about the value of a particular model organism and what it's doing in research, we're asking questions about representational scope or target in relation to phenomena or mechanisms, and then focused on or with respect to specificity or variety. Now, the other dimension, the practical dimension, manipulation criteria, oftentimes very mundane things, but actually quite crucial things: organism availability, costs of initiating inquiry, possible experimental techniques that can be applied, how quickly you can produce data and results. And of course, those are intertwined with the previous criteria about representation. And in some cases, they exhibit a trade-off relationship. So a model that faithfully represents organisms of interest might actually be difficult to experimentally manipulate, or an organism that's easy to manipulate might represent a general phenomenon of interest poorly. So in thinking about representational and manipulation criteria in choosing model organisms, there oftentimes is explicit attention to those sorts of trade-off relationships. And they can be weighted differently depending on the aims. But also it's important to highlight that some of these things are historically conditioned. So choices of organisms can be kind of locked into communities whereby people have been using them and they know how to use them. But that doesn't mean that when you try and use them for something different, that they might be easy to use for that kind of purpose. And in fact, keeping that in mind is really important because almost all of us inherit our model organisms. We start working on them during graduate school because that's what the laboratory we're involved in works on, and in some respects may not even be aware of how the model was chosen in the past. And so we can just see that with a classic example from fruit flies, which are a workhorse of model organisms in modern biology. And just to kind of make it concrete, Drosophila has shaped research agendas in sexual selection and immune response studies. But its particular mating system and immune architecture constrain the kinds of questions that can be addressed. Because those are the way that system works. And so they shape and structure the kinds of inquiry you can do. So traits absent in this model, such as alternative reproductive strategies or different types of defenses against pathogens, are often neglected because they lie outside the framework afforded by the model. And the important thing here is to not see this as somehow a reason to reject the model, but rather a reason to recognize that the model shapes the inquiry, and that we need to keep that in mind and recognize that in the context of our own models. Now, I mentioned at the beginning that it's not only the existence of model organisms, but actually a number of different ways that models are utilized. So sometimes when biologists talk about model organisms, you can get the impression that sort of all models are sort of model organisms. And then maybe sometimes people might say, I work on a non-model organism. But in fact, we can make some important distinctions in this space, right? We really can capture the distinct ways that biologists are actually working with models, and that in fact recognizing that heterogeneity is important for understanding why they're used in some contexts and not others, and sometimes why models don't do as much as we want them to do for us in investigation. So a really basic distinction that kind of captures a lot of the way I think the landscape of model organisms is in terms of standard models versus Krog principle. So let me start with the standard model. These correspond to the sort of NIH list that we've just seen when people talk about the mouse or the fruit fly or C. elegans. This is what they have in mind. Typically, you can study lots of different properties of organisms. And that's one of the reasons why these are preferred, because they kind of have this multi-purpose structure, as well as the ability to be investigated quite deeply in terms of causal mechanisms. And oftentimes the results are generalized quite widely. They also allow for manipulative requirements. So you oftentimes see quite sophisticated genetic analysis and successful standardization of these models, such that you get stock centers and other kinds of support infrastructure around them. Krog principle models are more of the, this is the right tool for the right job. And these have been really important in the history of biology. Auguste Krog is the one who originally articulated this principle. For a large number of problems, there will be some animal or choice, or a few such animals on which it can be most conveniently studied. So in the history, studying fertilization in sea urchins or electrophysiology using the giant squid axon are good examples of this. In the case of the giant squid axon, it was the comparatively large size that made it possible to reliably apply voltage clamps. And if you could reliably apply voltage clamps, you could actually make measurements, which was not possible with smaller scale neurons. So the experimental organisms are chosen in this case to answer specific questions, which means you typically have a very specific phenomenon as the representational target, and the results are typically generalized or extrapolated more slowly. And these organisms are not as standardized and sometimes less suitable for many forms of genetic analysis or for standard experimental tools. And sometimes it's also just because it's difficult to house them. So a lot of the work on sea urchins or even the giant squid axon were done in proximity to marine research stations where you could get easier access to that. But studying sea urchins in the sort of upper Midwest, where they have to be maintained in seawater tanks, is quite difficult and therefore is less portable as a research organism. Another distinction that is important, especially in thinking about medicine, is the difference between exemplars and surrogates. So exemplary models correspond to what we might expect of the fly and the mouse, where you're trying to exemplify something in larger systems. As the Nobel laureate Christine Nusslein-Volhard said, we study flies and frogs as examples for the development of animals in general. But of course, many people are interested in surrogate models because they want indirect experimental access to mechanisms for otherwise inaccessible phenomena. So mouse models of human psychological disorders might be examples, or mouse models of, say, alcoholism or different kinds of physiological conditions. And here the clear issue is the inability to experiment on humans and the need to find some other way to get at those questions. Now, there's even more kinds of model organisms. I don't want to sort of dwell on this, but I want to just emphasize that the range is quite wide. So in some areas of evolutionary biology, model taxons are used, so whole clades, where you're investigating interesting features across a clade and trying to understand the different ways in which developmental, physiological, or ecological phenomenon are occurring in that clade. Model life histories, where people are interested, especially in larval forms, and trying to understand their functional and morphological properties. And importantly here, these are not model taxa because they're not clades. And oftentimes the generalizations are made across very divergent divergently related and phylogenetically distant groups. And then finally, an area I've been interested in because I've worked a lot on EvoDevo is ways in which EvoDevo models are trying to kind of combine developmental modeling and evolutionary modeling to address specific questions like the origin of novel traits, the evolution of development, or the influence of developmental processes on evolutionary mechanisms. And just to kind of illustrate that, right, you can see here how some EvoDevo biologists have tried to use the cnidarian nematostella as a sort of comparison for the origin of axes in the bilaterian animals, and trying to understand to what degree the axes that are specified developmentally in nematostella do or don't correspond to the axes and the mechanisms underlying them in other organisms, like represented here in the mouse. So there's a lot more one could say about model organisms. A lot of people have said a lot of interesting things about it. For those who are interested in reading more, lots of historical and philosophical information about sort of how people have made these decisions, case studies, where you can see that in action. Excuse me. But now I want to transition to the second half of the talk. What is generality and why is it important? So let me begin with a misleading comparison. That misleading comparison is between physics and biology. So a lot of people might characterize physics as seeking universal generalizations, scientific laws, and that's how they explain, because they can appeal to laws, and their theories are mathematical systematizations based on those laws that give an account of how the phenomena operate. And so here one can think about the three laws of Newtonian mechanics and law of gravitation, or other areas that would exhibit that. And this would look very differently, I think, than people would see biology, where the discovery of mechanisms is foregrounded, that understanding the mechanisms is how we understand explanation, not in terms of laws, and that oftentimes theory is a proliferation of multiple molecular models, sometimes mathematical, sometimes not mathematical, and that this would make it seem like physics is interested in generality, biology not so much. They're just different sciences. They're engaged in different kinds of inquiry, and we just need to recognize their differences. Now, partly this is a misleading comparison that emerges out of philosophical discussions in the 20th century. Because for most of that time, people focused on physics as the exemplar of science, the way science should look, and therefore thought laws of nature should play a central role. All the generalizations were analyzed by some notion of universal generalization or law. But more recently, there's been a lot of interest in non-universal generalizations, categorizing them in terms of different parameters, trying to understand how they're invariant across some set of specified interventions, or maybe ways in which we can distinguish between generalizations based on materials versus those based on, say, design constraints. So the key upshot of all of this for our purposes is that biology can and does have generality as an aim of inquiry, independent of there being biological laws. You don't need biological laws to think about generalizations in biology. And that just because generalizations in biology might have many exceptions doesn't mean we aren't interested in seeing how general the claims that we make and discover based on our studies can go. Now, beyond the question of what generalizations are, we might be distinctly interested in understanding why. Why pursue generalizations, and why do that researchers structure their inquiry around different strategies. And if we're characterizing different generalizations, we can get better insight into what's going on when scientists reason the way they do. What's the epistemic structure of that? What kinds of standards of adequacy are being deployed for what counts as good, what counts as bad? That's the normativity. And even explicate differences in what is allowable as an inference, what counts as a good explanation or not in different areas. So let me give an example of the common tension that recalls the misleading comparison we just did between biology and physics. And that is a common tension between generality and detail. So if we're thinking about the cell, right, we can realize that there are ways in which we sometimes value general statements about cells, and sometimes we value increased detail about cells. And that in fact, these kinds of things are often in a trade-off relationship among those doing different kinds of inquiry. And so we'd like to understand how generality and detail are related in different scientific research projects. And characterizing generalizations actually can help us answer this question and understand, oh, well, the reason why you're going after that particular kind of generalization is because you are trying to abstract away from, say, details of a system to get that kind of generality. So there's three basic characteristics of generalizations that we can spend some time on briefly today. One is the scope, the range of things to which a generalization applies, the conditions under which a generalization holds, and the aims of a generalization, why we are trying to do it. So the scope can be broken out into a number of different dimensions. Clearly, one thing we're interested in is how our generalizations apply across different taxa. A second thing that we are interested in is how maybe those generalizations apply across component systems. And then also an interest in how they might apply across scales, especially levels of organization in biological systems. In addition to the dimensions of scope, we can also think about the conditions and the aims. The conditions relate to what we might call conceptual conditions, the research questions or problems that are being pursued, as well as the material conditions. Sort of under what conditions are we trying to generalize? Are we working with an in vivo system, an in vitro system? What kinds of ways have certain kinds of variability maybe been eliminated in the standardization of the system or by bringing it into the laboratory out of its original context? And then in terms of the aims of generalizations, we might be interested in describing a system better, explaining a system, making predictions out of the systems, or even some kind of control or intervention to change the system. And of course, these are not mutually exclusive. Now, just to kind of narrow this very specifically to explanatory generalizations for the sake of time, right, we can see that we've got a difference between mechanism generalizations, where we're interested in those cellular and molecular interactions, and principal generalizations, where we are trying to get at how relations or interactions can be instantiated by very different entities in different biological systems. And so what I want to do over just the next couple slides is try and very quickly show you what I did in a recent paper with former graduate student Yoshinari Yoshida. So a mechanism generalization could be something like thinking about BMP signaling and axis formation, right? And you can see here a kind of a cross taxa generalization for BMP signaling and dorsal ventral axis specification in a variety of different model organisms. We also could be interested in BMP signaling across component systems in, say, the body, in this case, the different places where the BMP signaling pathway is activated. Principal generalizations, by contrast, are not interested in the molecular details, but actually in this kind of abstract activator-inhibitor kind of interactions that can be mathematically modeled and in fact apply across taxa and component systems, as illustrated here in a number of different patterning phenomena during development, as well as across component systems, say in the palate or in digits or in left-right patterning for the entire body. So these distinctions matter because they direct the activities of reasoning differently. Mechanism generalizations focus on specific details that are widely conserved, but the principal generalizations abstract away from them. They have different kinds of warrant. Mechanism generalizations are based on evolutionary conservation, whereas principal generalizations are typically based on mathematical rules of some kind. And then they also have different scale patterns. Principal generalizations often apply across scales. Mechanism generalizations don't generalize as well across scales, in part because they depend upon certain kinds of interactions at scale. Now, they're also associated with different advantages and disadvantages. Clearly, there's many experimental techniques to get at molecular types and their activities, and that we know conservation means there's going to be able to be some extrapolation from what we discover. However, anybody who's been involved in characterizing mechanisms knows that it is incredibly hard work to get at those different interactions, to be able to confirm and have sufficient evidence for the role that a particular entity plays in a molecular mechanism. Principal generalizations, by contrast, are oftentimes established by how possibly explanations that you use mathematical tools and computer simulations to sort of demonstrate, excuse me, that a principle can recapitulate the phenomena. However, showing that a principle is instantiated in a biological system is often challenging because in many cases, more than one will apply. And so then how do you decide between them in terms of how actually, and that can be quite difficult to do. So to kind of wrap up the discussion of generalization, the important feature I want to highlight is that there's just lots of things going on. Traditionally, when people thought about laws, they just thought about how widely does this apply in space and time. But in fact, what we've seen is that generalization scope can be characterized in terms of dimensions, some of which are codified by the choice of the model organism that we saw earlier. So there's really no such thing as a scope of a generalization simpliciter, and the diversity of generalizations leads to different forms that are guided by distinct practices, and being aware of those practices helps guide what we do and why. So now let me turn to the last part of the talk, sort of thinking about this more in the context of our own model systems. Because clearly, a danger that all of us face in specialized research is that a focus on one or a small set of models can kind of insulate us, and we kind of end up talking to the same people about those models. And in fact, if you just look at the EvoFunPath website, right, clearly that's part of the whole rationale, is to focus on mycologists, to focus on fungal diseases, and clinical and environmental pathogenic fungi, and only to some degree the way other microbes might be relevant in some of those characteristic patterns of interest. Now, of course, none of that is to say these things are bad or that you shouldn't be pursuing them, but they kind of show the natural lines of boundary that exist between how one thinks about what one is working on and how general what one is working on is or is relevant to. Now, an important thing to highlight is that microbes have actually been models in substantial ways in the history of biology. So this is explicitly discussed by Maureen O'Malley in chapter 6 of her book, Philosophy of Microbiology, in highlighting what others have said, like bacterial genetics has had more profound impact on the development of modern biology than any other biological discipline. Hard to believe that E. coli would do that much work, but in fact, I think there's a lot behind that kind of statement. And if we look at the history, we can see that microbes have played distinctive roles as model systems, oftentimes in work that won Nobel Prizes, in all of the cases illustrated here, and also for particular areas of study, experimental evolution, ecology, development, and even to some degree cognition in more recent work. Now, we might then say, okay, thinking about our own fungal inquiries, how can we ask questions of that in light of the discussion about models and generality? So we might ask, what kind of model are you working with? How was it justified historically? How is it justified now? How does it fulfill representational criteria? Do you know what the scope is? Do you know what the target is? If you talk about that with others, do they agree with you? As well as other kinds of distinctions in the representational criteria, as well as their interaction with the manipulation criteria. Is there ways in which the system you're working with is lacking something, or has been discovered recently to not have capacity to be able to say, do certain kinds of experiments that you would want to do, or are more challenging than they might otherwise be? What kind of model is it? So in thinking about your model, what are you wanting your model to do? Do you want it to be a stand-in for something that's not otherwise studyable, then it's a surrogate? Or do you want it to be representative of, say, how fungal pathogens operate? In that sense, it would be more of an exemplar. We're trying to make sense of what kind of model am I working with? And then what kinds of generalizations are you seeking? What's the scope? Are you interested in different component systems, cross scales, cross taxa? What are the conditions? What questions are you asking? What laboratory conditions are pertinent? What are your aims? Are you going after mechanisms? Are you going after principles? Are you going after both? To what degree can you make these things explicit? And then, of course, how does your model facilitate identifying and confirming those generalizations, and how well matched is it for achieving this goal? Some practical recommendations, in addition to simply asking questions of your inquiry, that I think can help spur this kind of reflection and thinking on our research, is simply to ask more questions about the choice of model system. How exactly is your model a model, and have that be an open part of discussion. Ask more questions about generality. Does what I'm working on apply to other systems? In what way? If it doesn't, why not? And of course, a lack of generality can be interesting also. We're not interested in generality for this reason. You can actually interrogate the literature, say in a journal club, past or present papers from the perspective of these questions. Now, is there a shared viewpoint on what kind of general knowledge is being pursued? Are there changes over time? They're thinking about it differently 20 years ago than they're thinking about it now. Why is that the case? And then sometimes, at least, read papers outside of your own literature with these questions in mind. Can you figure out what others are up to and why they are similar or different to you? And if not, who could help you do so? Potentially inviting guests to join you for the conversation. So let me just point to one concrete example here. A recent paper I saw in PLOS Biology, very short perspective piece by John Drake. Why are there so few pathogens? Ecology and evolution and pathogen emergence. And an interesting thing that occurred to me in preparing this talk was that this discussion is primarily about why there are so few pathogens for humans. And an interesting question would be, how would the analysis change if generality scope dimensions were considered beyond, say, humans? And that seems like an interesting EvoFunPath type question, and the kind of thing that might be profitable to wrestle with. A few more practical recommendations. Utilize controversies to work through these questions. So sometimes it's the case that controversies might be disagreements over generalization, and differences over how people understand what their model is supposed to be doing. So the controversies themselves can be good gateways into these discussions. Tell your origin story, or ask somebody else to tell theirs about how they got to study this model organism, why they thought it was a good approach. Have they always been convinced they made a correct decision, or have been times of doubt in their use of the model and what that doubt was based on? Potentially host outsiders. Ask someone to visit your lab group or join a journal club who comes from a very different vantage point. Ask them what they make of your discussions, what they understand, what they don't. And just in the sense of meta-reflection here and self-promotion, philosophers can actually play a role in this. They're genuinely interested in what biologists think and do. And so, kind of like today, you could invite a philosopher to visit your research group, or even maybe join as an external member of a dissertation committee and help expose trainees to questions rarely asked by those in the field. Now, I want to close by talking about potential payoffs, because you might be thinking, look, that's all well and good, but I got a lot of work to do. I got to get back to the kind of main thing. So I think some of the potential payoffs for this kind of reflection include a deeper understanding of what you and others are doing and why, And you might be surprised in conversations with others to learn that the depth of understanding is not as deep as you might think, and only is surfaced when you are actively talking about it. You might come to be aware of novel methods and experimental strategies that are being utilized in other approaches, other models, other conceptual or material conditions that people are pursuing in their generalizations. You can also recognize neglected questions and overlooked areas that hold potential for making a significant impact, both within your area or across areas. Some of the greatest scientific impacts come from questions that are answered by means of connecting things across domains rather than only within one. You can come to an appreciation of alternative perspectives to scientific inquiry, some diversity within science. So recognizing, oh, wait, they're doing something different than I am. And why are they doing it differently? And trying to recognize and understand what that pattern of divergence in their approach is. And then finally, I think greater facility in communicating your research to a wider audience, who most likely don't share your generality commitments or your commitments about your models. And of course, this can be extremely helpful in the context of grant writing. So just to review very quickly, we talked about why model organisms are prevalent, many different reasons for that, and the different kinds of models that are utilized, some questions we might ask of them, and what is generality, why it's important, and then finally, how can we seek and find generality in the context of our preferred models and why we might want to do that and some of the payoffs that come along with it. So with that, I will stop. Thanks for your attention. Special appreciation to Matt for the invitation to join you, and I look forward to your questions and the discussion.
Thanks so much, Alan. I really appreciated that. Enjoyed it. Okay, so as I think we've described to you, the typical format is we have a few questions with the whole audience, and then everybody who's not a fellow leaves, and the fellows can ask you additional questions. I imagine, I mean, I'm really glad where you went in the end, because I was thinking along the way, how am I going to set the fellows up to be able to use that time well? But I think you just did that very much. But I just encourage all the fellows, try to have a conversation that brings in your own study system so you can wrestle, personally with some of these ideas in the context of your own research project. Okay, before we leave, or the professors leave and so on, let's have a few questions.
Hi, yeah. Old fellow here, guess not a fellow anymore, but I have a not so much question, but I'd like, well, first of all, great talk, super thought-provoking. And I was wondering about how all this discussion about model organisms ties into sort of modern day synthetic biology and using different organisms as like, platforms to produce certain molecules, or, platforms to do all sorts of things. I remember one of my profs used to call them like chassis organisms. And I wonder if I can get your thoughts about how this ties into model organisms and this whole discussion.
Sure, great question. I think I would sort of approach that through the lens of a particular aim of inquiry, which is control, right? Where in a lot of these cases we want to do something. We want to create some molecule. We want some kind of process to occur in a regular way. And so we are sort of tailoring the model organism to do that kind of work for us. And so in many of the cases where that's true, you can see exactly some of these dimensions coming out. So in some cases, the ability to have that manipulation comes from having worked with a species for a really long time and knowing its genome really well, knowing how to manipulate it, knowing how to get genes in and out of the system to be able to change capacities. But in other cases, we get really interested because those microbes have special properties like petroleum eating, right? And it's like, okay, because that microbial system does something really distinct that we're interested in, we really want to figure out how can we leverage that to accomplish these particular interventions. So I think many of those same considerations bubble to the surface, and what you're seeing there is a foregrounding of that aim of control and trying to do something with those model organisms.
Great, thanks.
I thank you so much for that really thought-provoking talk. There's a lot of food for thought there. I did my PhD with Sally Otto and doing experimental evolution with yeast, and so I spent a lot of time trying to reconcile like the hyper-specific nature of each lab experiment with like the goal of trying to find these generalities. And so I'm wondering if you can, I mean, especially when like replication of a single experiment may not produce the same results when done by different labs and so on. Can you talk about the relative importance of replication of like exact experimental conditions, using different conditions, using different model organisms or different or closely related organisms to the model, and like those different levels of kind of replication?
Yeah, this is a really, really important question, and one that I think gets at the heart of this, because when we are concerned about replication, we're oftentimes concerned about generality. It's like, okay, it might work here in this place, but does it work elsewhere? Does it work more widely? And is it somehow fragile to this setup that we've created? Because as we all know, you work hard to get your experimental system to do things for you. But that takes time, and by the time you get that working, it doesn't look the same way as it might look in somebody else's system. And of course, that's part of the reason why a lot of people emphasize standardization in the hopes that standardization will help in terms of replication across different domains. But obviously, if we discovered in experimental evolution in yeast some particular principle for, say, competitive exclusion under conditions of, say, resource scarcity, right? We really would want to know not just that it's about yeast, right? That it would be kind of uninteresting if it was just yeast that we were ultimately interested in. But in fact, what we're interested in is something more general, and the yeast are our strategy because they're more easily manipulable to do those kinds of experiments. But of course, you have to ask, well, what licenses that work to be generalizable? And so I think that sort of replication, stability under different conditions, oftentimes those things are a core part of that. But also, I think simply pursuing replication outside of the system of interest, sort of working with multiple systems and not relying solely on a single model organism. So just a quick example, I collaborator here at the University of Minnesota, Michael Travisano, in the Ecology, Evolution, and Behavior program, right? He actively maintains multiple microbes in his lab that people are working on, in part because each of those microbes are giving different kinds of information, and he's able to ask questions back and forth across them exactly of this kind about replication, stability, differences. But of course, it's a lot harder to maintain multiple organisms in a lab than it is to just maintain one primary. So I think that is part of the challenge of generality, is that generality does sometimes demand certain forms of scientific work that do sort of push us beyond some of the I'm just focused on my own organism situation. Aliza?
Yeah, thanks for a great talk. And I'm worried this is going to turn into more of a comment than a question, but I'm going to be that person anyways. And it is sort of a philosophical problem because I think, we're all pushed to write these big high impact papers where we say like, we've solved science. I've done this experiment and I can generalize it. Here's why everyone else should care. And as I started my lab, we've become really interested in sort of digging into one species. So we use Candida albicans, like many people here do. We've looked across many different isolates. And so just within one species, if we look across the phylogeny and we cherry pick isolates to try to represent the intraspecific diversity, our results are almost always never we can generalize with this one species. It's this isolate does this, and this isolate does this, and this isolate does this. And yet scientific society is pushing us to make big generalizations. And yet when we start layering on these sort of, nudges in our experiments where we vary different factors or use different isolates, never mind different species, it seems really uncommon to find any pattern that is generalizable. And so I don't know what to do with that.
Well, so let me take your comment as a platform to say something in connection with one of the questions I raised, which is we should be interested in why generality doesn't obtain, right? So clearly there is excitement when a conserved mechanism is discovered to operate in Drosophila and mice. People go, wow, that's exciting, and it is exciting. But of course, it's also really interesting if I'm working with a single species and among individual isolate lineages of that species, I'm getting all kinds of variability within that one species. Well, why is that? That in and of itself kind of becomes the interesting phenomenon. And I think that's something that we need to sort of really be good at emphasizing, that the bug is actually a feature, right? That instead of saying, shoot, it's not as general as they want it to be or something like that, we can flip it and say, you know what, this is not general, and the fact that it's not general is really interesting. Because if what we're seeing in the lab shows this much variability, think about how much variability there is out there in individual lineages for a particular species. In a way, you kind of get to generalization through a back door, because you say, well, look, if we get this much variability within a species, is that the expectation we should have for all species? That all species are likely to harbor this kind of radical variability, such that experiments within the one lineage can raise tricky reproducibility issues. And that is an interesting now claim about biology. But it definitely cuts against the grain of some of the hopes for the high impact paper, and what it would or wouldn't say. I totally agree with you there.
Okay, super stimulating, Alan. I think we've all got lots to think about. It is at the time when we should transition. I'll leave it up to one of the fellows is going to now moderate the discussion to whatever extent they need to. And I don't
Sorry,
remember.
Matthew. Oh, there's only one question in the chat.
Oh,
Is
yeah,
that
Do you
one?
want me to read it? Huh?
This is curious how the standard is defined.
Yeah.
What's the standard? The standard for, is that for reproducibility or replicability, what we were talking about a few minutes ago? Okay.
Yeah.
So that actually is a really good question, because oftentimes that standard is not always defined in advance. And so people will sort of discover that's not replicable, but then, well, what would be replicable? And then a conversation needs to happen about what exactly is the standard. So I think that's really important, because sometimes people think, oh, we all agree on what would count as replication. And in fact, that's not true. And it only comes out when people start actively saying, well, what is it the case that I expect of my experiments, and under what conditions is variation across isolates or across species exactly what you would expect? It has to be brought to the surface and discussed explicitly. It's a good question. Okay, well, thanks for all the faculty who were here. Appreciate it. And I look forward to the conversation with the students and trainees.
So which fellow is
Thank
leading?
you.
I'll lead the question period.
Okay, thanks, Penny. Alan, thanks so much.
Thanks, Matt.
Bye. Dr. Love, do
Okay.
you need to take a little break or are you good to go? And
I'm good to go. We can just keep going. So
okay, so I'll just stop the recording quickly.